Good evening and welcome to our science lecture this evening. My name is Hank Fitzgerald. I'm the Director of Original Programming here at the Wayne, so welcome to the Wayne on this Tuesday night. Wonderful to see all your faces. So tonight we have Insects of the Shenandoah Valley. Next Sunday, January 24th at 4 p.m., we have Cattle in the Valley. Insects this week, and then at the end of the week we have cattle. Isn't that fabulous? So if you have any youngsters that are that are interested in theater, we have Studio Wayne virtual classes starting. So please check out our website. We have voice, we have makeup classes, movement, dance. So please be sure to check out our Studio Wayne if you have any youngsters that are willing to check out online classes, which are virtual classes right here at the Wayne. We have a benefit concert series with our Monday night movie host, Jimmy Overton. So those who like Jimmy Overton, at the end of each month, we're going to have a concert series of 50s, 60s, and 70s, all the way up something different each month. So please check out our website for that. And then the February 13th, we have a Sondheim concert coming up. We have some actors coming in from New York, professional singers. So please, if you have that Valentine date scheduled, be sure to come out here to the Wayne. We'd love to see you. Okay, now I'll hand it over to you.
All right, thank you very much. Good evening. I'm Joe Kuiper. I'm the director of the Virginia Museum of Natural History. And what you have right here is, of course, a map that shows where your Museum of Natural History is. So if you look down to the lower left, you see Roanoke, and you see Blacksburg, where Virginia Tech is, and so forth. And that teardrop green shape there shows right where your Natural History Museum is. So we're in a really cool part of Virginia. Our curators love it, and you're going to hear from one of them tonight. But that being said, we are a bit removed from a lot of the Virginia population. So why are we here in Waynesboro? Well, thanks to the local nonprofit Center for Cold Waters Restoration, and thanks to the South River Science Team, we have been working to integrate into this community so that we may open up our first permanent branch of the Virginia Museum of Natural History right here in downtown Waynesboro, just about a couple hundred yards downhill from here next to Constitution Park. So we've been working very hard on that. This lecture series tonight is kind of part of our connection to the community, and I want to thank you all for coming tonight, and I also want to thank those people who are joining us online. So the Virginia Museum of Natural History, there's our kind of front porch right there with our Diplodocus, a large long-necked dinosaur from about 150 million years ago, adorning the front of our building. We want to bring what we have there in Martinsville, which has our scientists, it has our educators, and it also has our exhibits. We want to bring those programs right here to explore new ecosystems that we don't have down in the foothills of the Blue Ridge. We want to bring our education programs, and we want to bring some of our collections. We have 10 million specimens and artifacts in our holdings. And of course, you can't get all that on exhibit. You can't make it all publicly accessible. But what we can do is create a branch with public exhibits, allow people to come and visit and learn more about the natural environment. And the branch that we build here will be place-based. It is to talk about the stories of natural history right in this part of the Commonwealth. Here we go. So what we've been doing is over the past several years, more than that now, 8 years or so, we've actually been doing a few analyses, trying to figure out what is going to be the best plan of attack. And just a bit over a year ago, we've completed what the state calls the pre-planning process. And here you see Constitution Park to your lower right, and the South River is just a little bit farther to your lower right in that image. Right here in downtown Waynesboro. And this kind of shows an aerial view of a concept of what our building would look like, a two-story building that is representing, roughly speaking, around 30,000 square feet total, the majority of which is for public use. And so we really look forward to serving more people that way. The next step in the state is to do what's called detailed planning, which is a regimented 18-month process, and we're ready to do that. However, with COVID, pandemic, and all that kind of stuff, we, of course, are slowed down a little bit. We're going to just have to wait until the funds that have already been allotted for our detailed planning, so we're in the state budget, they're just not going to give us the money right now. Makes sense. They have to pay the bills to fight the pandemic. So hopefully within the next 6 or 18 months, we'll just have to see how things go, that we'll get that portion funded and we'll move along, and then we get to the point where we'll do the plans that you hand over to a contractor to actually start construction. And so, what we did during that process was to create the concept of the building that we see here. This on the right is kind of as you're coming across the Main Street Bridge over the South River, and you see the museum there on the right-hand side, right? But we also started to look at what the inside might look like. So this kind of shows a schematic of the inside of the second floor, the top floor of the building. And we're showing this particular one because it relates to our talk tonight about biodiversity, the insects of the Shenandoah Valley, and an entomologist perspective on those. But the vast majority of the square footage here is, again, going to be public exhibits. And this kind of shows it fleshed out a little bit more. There's going to be stories about the geology of the region, the hydrology, the cultural anthropology of the region, but also things like the zoology, the animals, the botany, the plants that live here. And that's what we're going to be listening about tonight. So what I would like to do is to introduce our Associate Curator of Recent Invertebrates, Dr. Cal Ivanov. He's been with the museum for 6+ years at this point, so it feels like we just hired him yesterday, but he's been around for a few years now. He and the other curators that work at the museum, they are going to be instrumental in developing those stories and working with local partners at our universities and our nonprofits to talk about what those exhibits might look like. So tonight, Cal's going to talk a little bit about his expertise and talk about how that relates to this particular fascinating region of Virginia. So without further ado, let's give a warm welcome to our curator, Dr. Cal Ivanov. Cal, come on up.
Thank you, Joe. And thank you, everybody, for coming tonight. I was here in this same building about almost exactly a year ago, just 2 days shy. And things were quite a bit different back then. There was, of course, a lot more people around, but hopefully a lot of those people are joining us online. And what I want to talk about today is, of course, touch up on our plans of bringing the Virginia Museum of Natural History to Waynesboro, but also talk to probably one of the most biodiverse regions in the entire nation, not just in Eastern North America. So I'll start with something familiar to all of you. We've all visited museums at some point of our life, and feedback from visitors to the Virginia Museum of Natural History and many other museums indicate that these people come to these facilities because they feel welcome. They're having fun, and ultimately they learn something new. And probably the most familiar aspects of museums are the museum's exhibits. But museums as institutions are way more than that. Museums exist because of their collections. Collections are at the heart of these institutions, whether we realize it or not. But what are collections? Every single one of us at some point of our life probably has collected something. In many cases, something that probably made sense only to ourselves. Why do we do it? Out of curiosity, having fun, bragging rights, possibly. But museum collections are not stamp collections. Collecting is not the goal. We collect these specimens and artifacts because they provide information. And every single one of these specimens can tell a story. And every single specimen can be examined by a different researcher, a taxonomist, an evolutionary biologist, biogeographer, and they all will find that same specimen valuable for a different reason. These specimens, these collections are especially important today in the times of very fast ecological and climate change. We can use these specimens, these collections, to recover the ecological conditions of the past. We can gather, we can harvest a lot of information. These collections also serve as our bedrock, our base of taxonomy, the science of naming and classifying living organisms. In addition to that, the world's natural history collections harbor an estimated 3 billion specimens. They reflect what we know about the world's biodiversity. How many species inhabit our planet is probably the most fundamental question in science. The answer to this question is still enigmatic. To date, there are approximately 1.9 million named species, and that includes everything from bacteria to plants. However, the estimated number of taxa is probably way higher. What you see here, you don't have to try to read all the text, but a study published a few years, about a decade now back, estimated that there is roughly about 8.7 million species of eukaryotic organisms only, right, excluding taxa such as bacteria and archaea. Give or take 1 million species. That means despite about 250 years of studying the world's biodiversity, we have but scraped the surface. And again, when you think about this number, think eukaryotic only. But what about bacteria? What about archaea? Only a few years back, researchers from Indiana University attempted the unthinkable. Using laws of mathematical modeling, mathematical scaling, they estimated that the world is home to anywhere between 100 billion to 1 trillion species of prokaryotes alone. Only about a little over 5,000 of those have been named. So there is still a lot of work to be done. The Virginia Museum of Natural History is a fairly young institution. It was formed as a private organization back in 1984, became a state agency or institution in 1988, but it has taken on a leading role of studying the biodiversity of the Commonwealth. Since the time it opened its doors, 16 curators in a variety of different disciplines have lended their expertise, their skills to studying Virginia's biodiversity. And they have left an impressive legacy. The work of the late Dr. Hoffman, the work of the current curator of mammals, Dr. Nancy Moncrief at the bottom left, the former curator of archaeology, Dr. Elizabeth Moore on the top left, have built one of the most impressive collections, not only in North America, but in the world. The Virginia Museum of Natural History harbors one of the finest millipede collections in the entire world, probably in the top 5. And that is almost solely due to the work of the late Dr. Hoffman. We have one of the finest, actually the largest collections of tree squirrels in North America due to the efforts of Dr. Nancy Moncrief and colleagues. We have probably the best zooarchaeology collection in the eastern United States. And that was achieved, again, only in the span of about 3 decades. Imagine how much more we can do. And these collections can tell stories. There's a lot more stories that they can tell. So back in 1998, Dr. Dave Smith from the Smithsonian Institution looked and recorded all the species from a small group of wasps that developed as egg predators in the eggs of cockroaches. There are 7 species known from eastern United States. He recorded 5 back in 1998. In 2011, the little snippet on the left, he published a 6th species that he previously was not able to record. And that they're highlighted in red. He guaranteed that the 7th species undoubtedly will be found in Virginia, although despite his intensive efforts, he did not manage to do so at the time. The specimen that you see up top is actually one of the first specimens that I examined and curated when I joined the Virginia Museum of Natural History back in 2014. Not that big of a deal because this is not an exceptionally rare species. The cool part of the story was that that specimen was collected back in 1937 in Richmond, in the Richmond Dairy Company. The building still stands near downtown Richmond. It was part of the University of Richmond insect collection. Unfortunately, the entire collection was mothballed. This university discontinued support for the collection. And if it wasn't for the Virginia Museum of Natural History, that collection would have been lost. Back in 1991, the entire insect collection was transferred to the Virginia Museum of Natural History, and that is how I came across that specimen. And of course, then I followed up with another short publication saying, yes, now we have that last species of this particular group, and the first specimen is in our collection. Speaking of collections, building collections takes time. It takes effort. From the time you go in the field and target a particular group, bringing the samples back into the lab, sorting through all these specimens, preparing these specimens, labeling these specimens, identifying, labeling them again, ultimately curating, putting them away. And that, of course, is related to every single specimen. The collection at the Virginia Museum of Natural History can be considered of moderate size, and we have more than 10 million items in our holdings. Larger institutions, such as the Smithsonian and, of course, the British Museum, have orders of magnitude more specimens. So imagine the amount of effort that goes. And that means that we have responsibilities. We have responsibilities to protect these specimens and ultimately leave them in the same shape that we found them. And once properly prepared, properly taken care of, these specimens can virtually last indefinitely. I already told you the museum is only about 30-some years old. Our oldest specimen, if you can read the label, is this giant leopard moth that was collected back in 1872. The label in the middle is the original label. The one on top was a label that was transcribed by the late Dr. Hoffman, the former curator of invertebrate zoology. So it is not only a historical specimen, it's a legacy specimen. It was collected by one of the most eminent entomologists of its time, John Henry Comstock. So if properly taken care of, these specimens, again, can provide information for many, many years to come. Unfortunately, if these collections lose funding, if they are not properly taken care of, they can quickly turn into a pile of unusable bits and pieces. This is pretty much what happens to any, in this particular case, insect collection after being mothballed for about 2 to 3 years. Specimens lost to negligence, to lack of support, are specimens lost forever, along with all the data that they can provide. We have responsibilities for these collections. And I've been talking about the curators, but museums are way more than that, as you can imagine. Our work would have not been possible by the help of great many other individuals. Everybody from those working on buildings and grounds at the front desk, at our fiscal offices, of course our board of trustees, our director, they all do their part in order to make our work easier and of course to make our mission achievable. All these individuals, we play hard, we party hard, we work hard. All of these individuals, along with our volunteers, interns, research associates, they're all devoted to the protection and preservation of the natural heritage. In this particular case, focused not entirely, but primarily on Virginia and the southeastern United States. And that, let me tell you, biodiversity is unmatched. Unmatched in the East, and very, very few places in North America can match Virginia in the diversity of landforms, of habitats, and ultimately animals and plants and single-celled organisms. Going from the sand dunes, the maritime forest of the eastern shore, through the pine and oak woodlands of the Piedmont, to the subalpine zones of the southern Blue Ridge, all of these are unique environments. All of these harbor an impressive amount of biodiversity. The southern Appalachians, which include parts of southwestern Virginia, have been recently identified as one of the 6 biodiversity hotspots in the United States. These are areas that harbor an incredibly high number of species. The closest one to us is the Florida Panhandle. If we want to see comparable number of species, we have to travel all the way to the West Coast or even to Hawaii. Virginia's Clinch and Powell rivers harbor the highest diversity of aquatic organisms in the entire nation. So anybody that focuses their studies on groups such as stoneflies, caddisflies, mayflies, they want to be in Virginia. And as a matter of fact, a great number of organisms have been described and only found in Virginia, including some that you probably heard if you were here last week, such as at the bottom there is, do you guys recognize, obviously, a salamander, but it's a salamander that is found not too far from here. That's the peaks of otter salamander, endemic to a very, very small area of the Blue Ridge Mountains. We have a number of other endemic, of course, salamanders in Virginia, and we have no less than 130 invertebrates, insects and their allies, that have only been recorded from the state of Virginia. Owing to its geographic location, to its topographic variability, diversity of ecozones, Virginia ranks as one of the most biodiverse areas across the entire nation. And there have been thousands of pages, hundreds of guides focused on the larger, the more charismatic groups. We have now an incredibly good account of the flora of Virginia, which was published back in 2012. Over 1,600 pages, over 7 pounds in weight, including detailed accounts of 3,164 species of plants found in the state of Virginia. We have a good understanding of the number of mammal species, 115. We have a very good account of the bird life, over 450 species of birds recorded from Virginia. Reptiles, 70 species. Amphibians, 87 species. Freshwater fishes, 210. But what about bugs? And that has been the million-dollar question. What do we know about them? Well, we know that we don't know that much. Based on existing lists, based on studies conducted in nearby states, Dr. Hoffman and colleagues postulated we should expect at least 20,000 species of insects alone in Virginia, and probably only about half of those have been recorded to date. There are some very striking exceptions, such as the very recently published Scorpion Flies and Allies of Virginia. That publication was published about a little over 4 months ago by the Virginia Museum of Natural History, and it is the first and most detailed account of the scorpion flies of Virginia. And Virginia, it turns out, is second in North America only to Mexico in the number of taxa recorded from the state. So many exceptions, a lot more work to be done. And despite a rather early onset of myrmecological studies in Virginia, ants are no exception, or they were no exception to that trend. So the very first accounts of ants from Virginia dates back from 1867, when Samuel Buckley described 3 ant species from the state. And we're talking about 150 or over 150 years ago now. But despite that fact, when I first arrived in Virginia, the state was only one of very few blank spots on the map of eastern United States in terms of our knowledge of the ants present in that area. So logically, very quickly after I arrived, we initiated the Ants of Virginia project. And that is a project that I will spend the rest of my time here talking about. What is that we do? What is that we found? And the 4 people that you see up top are primarily the people that have been involved directly with this project, including our director in the top right-hand corner, every time that he travels. And he used to travel a lot, except for that past year. So I'll send him with empty bags. And every time he stops, he'll collect a couple handfuls of soil and leaves and bring them back to the lab. And those couple handfuls actually contain a great amount of different critters and have produced some of the most fascinating finds, as you'll see. But when we started, of course, our first question was, well, what has been known to date? We have no single comprehensive list. Nobody has taken the effort to put everything together. But we went back. We went back 150 years and found every single source. We probably missed a few. It happens. But at the time, we amassed all the information, and we found that at the time, this is roughly late 2015, early 2016, that there have been 130 different species of ants reported from the state at one point or another. And that, unfortunately, was a rather severe underestimation of what we know. And how do we know that? Well, in 2012, an updated list was published for North Carolina listing 192 species. And frankly, based on what I already talked about, the diversity, the topography, the variety of physiographic provinces, Virginia should have probably as many, if not more, species. So obviously, there was a lot of work to be done. The next logical step was we saw what was published. Well, let's now go back through these collections and see what they have to offer. Pretty much is that example with that wasp, the egg parasitoid of cockroaches. Are there other specimens that are tucked in somewhere in a collection that we don't know about, that nobody has had the chance to examine? And logically, we started with the Virginia Museum of Natural History collection. And at the time, again, late 2014, early 2015, the entire collection comprised 907 specimens. That's it, 907 ants, most of them unidentified. Then we went to Virginia Tech, then we went to Radford University, and then, of course, we ultimately ended up at the Smithsonian a few times. In addition to that, in order to save on time and money and travel, of course, nowadays it's easy to go and examine collection just by sitting in front of your computer. A lot of museums, a lot of large institutions have completely digitized their records, meaning they have captured that information and they make it available for us. So we examined all those records. And simultaneously, of course, while we were doing that, we started our field work. And ultimately, our goal is, this is the map. Everything in yellow are places that we visited at least once up until the end of 2018. Since then, we have actually touched up on a few more of those blank spots. At every location, we collected a bunch of leaves and soil. Again, here is Dr. Kuiper, here is me collecting. And in addition to that, we spent time, of course, hand collecting and searching for a variety of different critters in those areas. So by the time of 2018, we visited roughly about 2/3 of all of the Virginia's counties and independent cities. And ultimately, what we managed to do as a result of our examination of museum specimens and our field work, we added 34 taxa to the Virginia's list, 34 species that were previously not known from the state. And as a byproduct of that research, of that field work, we increased the Virginia Museum of Natural History collection from 907 specimens to over 40,000 specimens, all identified, all curated, all data-based, and available to anybody that is interested. And we have probably roughly as much more still backlogged material, still sitting in the lab that we haven't had a chance to examine yet. But we decided it's time that we cannot wait any longer, so early in 2019, we published the first comprehensive list of the ants of Virginia, and at the time, that list contained 164 taxa. So nearly all of those 34 species that were newly found can be considered rare, because we have only found them by either a single specimen or maybe a couple specimens. And there is 4 main reasons of why a species may be considered rare, or why a species may escape detection. The first of those reasons is species may escape detection simply because these are either taxa that were recently described, meaning that they were not recognized in the past as a separate species, or even taxa that are still undescribed. What you see here is 3 different morphospecies that we know they're different, but they still have not been formally described. Therefore, very few people actually know about their existence. And one of these is very, very rarely collected species. And in Virginia, it's collected 3 times, 4 specimens, 2 of them from Mattaponi Wildlife Management Area in Caroline County, and 1 from George Washington National Forest. And these are the only specimens in Virginia. Another reason of why species may be considered rare is because simply they reach the edges of their distribution. The 3 species that you see on this slide are southern forms that are much more common further south. But they ultimately reach their limits, their tolerance limits in Virginia. And Temnothorax tuscalosi in the middle here, the northernmost known location of that species in the world is actually Indian Field Creek in York County. And that specimen was collected by Dr. Kuiper on one of his trips. Of course, the opposite is also true. Species may reach the southern edges of their distribution. What you see here is a species of socially parasitic ant that extends across the Blue Ridge Mountains, and ultimately its distribution ends near Tennessee and Virginia. So that's the second reason. The third one is species may be considered rare because they're confined to unique environments, unique habitats. Here you see a species of arboreal ant, or ant that spends most of its time above ground. But that species in Virginia is entirely confined to swamps. Cypress water tupelo swamps. That's the only area we have found it. And believe me, myrmecologists don't go collecting in swamps because you don't expect to find too many things. But if you don't go there, of course, you will miss some of these taxa. And the last reason is certain taxa are just hard to find. And that's why I said, again, it's very important. We use these tools that you see here, simply just cotton bags with a mesh bag inside. We load them with dead leaves and soil and let them dry. And as that substrate dries, the critters ultimately get driven and fall down inside these containers. And this is just a representation of one such sample, or the ants that were extracted. Some of these are things that are about 2 millimeters or less than 1/10 of an inch, or maybe 1/8 of an inch. And not only that, but some of them, such as many members of the genus Stromigenes, will play dead. They will play possum. So you're in the field trying to find something that is 1/8 of an inch, looks like the leaves and the substrate, and the moment you disturb it, it lays motionless. So it's very, very hard. So that technique actually allows us to collect many of these critters. Others are arboreal. They spend most of their lives high in the canopy. And we don't have that many arboreal species. If you are down in the tropics, there are techniques that have been developed, especially fogging, that you can knock species, but that's an expensive technique. It requires a lot of effort, a lot of labor. Around here, it's not worth doing fogging because the return will not be worth it. And that means we can miss some species. What you see here, that particular specimen is the only specimen of that species known from Virginia. These guys have a really interesting biology, but I don't have the time actually to go into this. I'll be glad to talk to you after. But that specimen I actually found right in front of our building, the Virginia Museum of Natural History in Martinsville, while I was taking my lunch break. Sometimes luck helps. Actually, a lot of the time, luck helps. And undoubtedly the most, or the rarest thing we found is one of these tiny Stromigenes taxa. Stromigenes memorialis was described back in 1998 on specimens collected in 1997 from single locality in Daniel Boone's National Forest, the Red Circle in Kentucky, in eastern Kentucky, never seen since. One of the rarest ants in the world. And then, back in 2016, again, Dr. Kuiper went. An even funnier story, I sent him somewhere, he got lost, he ended up in a totally different location, came back with a sample, and I'm like, well, let's look what is there. That specimen was there. 450 miles to the east, only the second known locality of that species in a totally different habitat. That tells you how little we know about these critters. But even if we manage to record every single species, that still leaves a lot of questions. We will still know very little about what these critters do. What is their lifestyle? What is their natural history? What species are pests or invasives? What are native? Which one needs protection? Don't forget, this is and will continue to be for quite a while a work in progress. Since we published the first account a few years back, we started finding new things. The two species on the left here, note they're actually all three, but note the small or almost non-existent eyes of these guys, the short appendages, the pale body coloration. These two species are subterranean, hypogeic. They rarely, if ever, come to the surface. So how did we find them? Well, we found them because we started another project with Clemson graduate student Kurt Harden, formerly a volunteer in the bug lab at VMNH. Then he moved to be collections assistant, now research associate. But he started this project looking at rare ground beetles in the soil using this very simple contraption. So trapping below the soil surface, down to about, in some cases, about a foot to foot and a half. A great number of interesting finds. And we're still working, actually, on that project. About 2 weeks ago, we went and collected the last set of traps. Not only are we finding new records for Virginia, we actually found our first undescribed species in Virginia. It was coming. There is no doubt there will be more. But ants as a group have been fairly well studied. If you study millipedes, the world is yours. You can find actually new species of millipedes probably just outside, probably in Constitution Park. I can almost guarantee it. With ants, it's a little bit tougher, but nonetheless, we found only 3 specimens of this undescribed species of Nylanderia, and all of them came from Fort A.P. Hill. I didn't collect the specimens, but people at the Department of Conservation and Recreation know I work on ants, so they send me these specimens, and yeah, it turns out it's something new. And it is the first new species of ant actually to be discovered in Virginia in over 60 years. And once formally described, it will be only the 9th species that have been described from the state. So for those lists that we create can help us elucidate certain aspects. When we published it, there were 15 non-native species or exotic species of ants recorded from Virginia. That's not the ants. Ant travel, people carry ants with them, still do, despite our efforts and the efforts of the U.S. Customs and elsewhere. And not surprisingly, about 4 months ago, we discovered another non-native species, the infamous little fire ant, not to be confused with the red imported fire ant. And that was in another backlogged sample, this time not collected by Dr. Kuiper, but collected by Bob Anderson from the Canadian Museum of Nature, a beetle expert. He collected some samples and he left us the bycatch. We're going through it and discovered one specimen in Lee County. Rather surprising because the rest of the introduced range is actually confined to the Gulf Coast states. How did this species make it there? Why one specimen? Is it established? If it gets established, that's bad news. Unfortunately, again, we discovered it fairly recently, so we will go there and we'll try to examine the area and see how this species ultimately has become or has not become established. And even for taxa that we know a lot about, the red imported fire ant. Virginia Tech graduate student Morgan Malone started work on the spread, among other things, of the red imported fire ant in Virginia. Despite current restrictions on travel and despite all the hurdles she had to jump through, he did a really, really nice job this year. And she actually added some data to our understanding of the spread of the fire ant. What it turns out is fire ants have nearly reached the city of Danville to the west and are already north of the city of Richmond. What makes it interesting, these areas were not predicted to have fire ants up until the year 2080. We are 60 years ahead of schedule. And what species need protection? What species are rare in Virginia? Special status? We have 7 taxa that have been included in the International Union for Conservation of Nature list of threatened taxa. But that list was last updated in 1994. So we certainly need a little bit of extra effort. And ultimately, the aspect that will take the longest to answer is, well, what is that these 169 now species that inhabit Virginia do? We know very little about most of them, maybe with the exception of those that concern our health, maybe those that concern our crops. The rest, we know very little. And in that regard, back in 2017, we started a project trying to elucidate on the natural history and the life cycle of one of those small, tiny, cryptic ants of the genus Stromigenes. And we picked the most widespread and the most common of these species because it was almost guaranteed that we will find it. Pretty much, they're hard to collect. The untrained eye will have a very, very hard time picking them up. But if you know what you're looking for, ultimately, I have no doubt they will pop up all over Virginia. But we still know that information that you see up top, the text on this slide, pretty much summed up everything we know about this species. So using some rather interesting novel techniques, actually using a technique we did not develop, we borrowed it. It was been used in the past, but we use these little blocks. You can see the size. They're only about a couple inches in length and maybe about 1.5 inches in width. We created them pretty much pieces of wood with some chambers inside, and we sandwiched them together and put them in the field. We created free space living for ants. And then went and picked them back again at the end of the season. They're already nice and seasoned. And then, of course, brought them to the lab and examined them. And ultimately, we found over the course of 2 years, we laid nearly 200 nests. We recovered most of them. We lost a couple along the way. But Stromigenes rostrata, the species we were interested, actually accounted for roughly 2/3 of the colonies of the ants that we encountered. So one job done. We knew they were in the area. Now that we knew that they have propensity to nest in preformed cavities, the next logical thing, and again, this is not a thing we came up with. It was fairly well known before, but we went and started examining natural cavities. And in the area, those are old hickory nuts or old acorns. The entire study, that second portion, was conducted in a relatively urban area. And this one right here is Dr. Hyper's home. He was kind enough to actually let us in his backyard. That has a fairly well developed old growth forest. We spent a lot of time there, and ultimately, we collected an additional 57 colonies. All of them came from an area that is actually smaller than this space here. So incredibly high abundance and density of that particular species. So now we had plenty of colonies. We could start tackling some of the questions we had. The first thing was, were there differences between the colony sizes in the artificial nest and those acorns? And turns out there are not. So we combined the data, and ultimately, we had good representation of the size of the colony, how many workers. They ranged anywhere between 3 and almost 160 individuals, workers, so fairly small colonies. Each colony had anywhere between 1 to 13 queens. Having multiple queens in ant species is not uncommon, but we'll get to that very shortly. And we compared that data to the only previous study that has previously attempted to estimate the size of the colony in Stromigenes rostrata. It was conducted in Maryland, roughly about 250 miles to the northeast. And what we found was kind of interesting. Our colonies were smaller, same species. Our colonies had fewer queens. And we were like, that is very interesting. And we went back through the literature, and of course, we were not the first ones to actually come up with this idea. Of course, when we talk ants, E.O. Wilson probably has discovered it before you at some point. So back in the 50s, he shared that idea that it seems that ant colonies, not just Stromigenes rostrata further north, actually have larger colonies. And it makes sense. Shorter season, More limited resources, you need more workers, right? Makes sense. But we managed to empirically show that with that particular species. And then, based on all of that data, we managed to actually, for the first time, reconstruct the life cycle of the entire colony. When are the eggs produced? When are the workers produced? What does workers do? When are the reproductives produced? And we found out that actually comes about end of October. They hibernate, but they hibernate as fourth instar larvae. The workers, the queen, and everything else ceases at the fourth instar. And they resume again development early in the spring. All the reproductives seem to be produced very late in the season. We found them inside nests up until mid-September. That means they start flying and mating after September, very late. But despite that knowledge, actually, and we tried 4 seasons in a row, we could not catch the onset of the mating flights. We could not catch these reproductives outside of the colony. We used a variety of different approaches, spent numerous hours in the field. So we'll try again next year. I mean, that's how things work. And I mentioned multiple queens. Polygyny is a term that is used by myrmecologists to note species that not only have multiple queens, but have multiple functioning queens. Imagine an ant colony may have 10 queens, but only one functional, only one laying eggs. Polygyny means that all 10, or at least some number in between them, will be egg-laying. That's a question we had no answer to. So what did we do? We went and started dissecting these individuals. We dissected over 150 queens, examined their reproductive status. So what you see here is a structure called the spermatheca. Ant queens mate once, or during one time of their lifestyle. Maybe with multiple individuals, but they store the sperm. That means that allows them to continue lay eggs throughout their life. For example, leafcutter ant queens can live for over 20 years, and they will continue to lay thousands and thousands of eggs. So we examined the spermatheca, and what you see inside are actually sperm, right here. So when we examine this structure, and you can actually see the spermatheca, this is the size of the spermatheca over there, we can say if the queen was mated or not. By examining the reproductive structures, these yellow bodies are actually remnants after an egg has been laid. So we can actually tell if that queen not only has mated, but also laid eggs. And based on these dissections, ultimately, we found out that, yeah, it is truly polygynous species, meaning that all of these queens, or most of them, actually mate and actively lay eggs. We kept some colonies in the lab. We observed them for over 3 years. That gave us also an indication of their behaviors, how they hunt their food, but also how long the queens live. And it turns out they're quite long-lived, something that we did not quite expect. And this is just, again, I can probably spend hours. I don't have hours. I'm virtually done here. This is just in summary to tell we spent about 5 years trying to elucidate on the life history of a single species. Now we have about 168 more. We may run out of time. But probably one of the most interesting things, and with that I'll conclude, is, and it's probably will be the hardest to test. So what our hypothesis currently is, how are these multi-queen colonies formed? Colonies are formed by one queen. After she mates, she excavates a colony and ultimately starts her own family. And then new queens are added. We have multiple line of evidence that these new queens are actually joining a previously existing colony and ultimately increasing the number of queens. How they find that colony, how they get accepted, how related are they to that colony that accepts them, we still don't know. Nonetheless, a very, very cool story, and there's a lot more to come in the future. But that's all I have for you guys tonight. Hope you enjoyed it.
Does anybody have any questions for the speaker?
Yeah, you talk about protected species of ants. What does that mean, and how do you protect them?
Well, it means like that species of Strumigenes that I showed you. Obviously, that's a rare thing. Means that it's not found in many areas, means there's very few individuals, so it's at a greater threat of being extirpated. And the only way that we can protect insects, not just only ants, is by protecting habitat. That is the only way that we can ensure that we're increasing the chance of survival. It's applicable to bees, it's applicable to beetles, it's applicable to millipedes the exact same way. But in order to say that something needs protection, we require a lot of work, right? If deer start disappearing and we're down to 100 deer in the state of Virginia, we will know it, right? They're large animals, we can see them. If we lose most of the African elephants, and unfortunately some of these trends are already present, we will know it. With insects, you can never be sure. That is why it's very, very hard to put an invertebrate on a protected list. Luckily, we have some success. The rusty-patched bumblebee, only a few years back, is actually the first bee in the continental United States that was placed on that list, simply because of decreasing numbers. It can work, it just requires a lot more effort.
Yes.
Most of the time, it's just me. If I'm lucky enough, I'll have one person with me. If I'm really, really lucky, we may get 2 or 3 people. Typically not large groups, and typically when we go into an area, we all disperse. You park here, you go do your own thing, because very few are focusing on the same group. Some will look for millipedes, some will look for beetles, some will look for flies, so we didn't congregate, but typically small groups, and most of that work was mostly by myself. Actually, I have not used Facebook. I try to actually ask and welcome specimens during a lot of talks, but that is absolutely a great idea. I always welcome specimen. Even if it's a picture that you can take and send it to me, all of these actually images or specimens can provide data that we need. So, yeah.
Cal, I know that your title is Recent Invertebrates, but can you tell us a little bit about the history of ants? How long have they been in the fossil record? And is that diversity related to how long they've been around?
Okay, so good question. So recent invertebrates, probably the only department I know in the entire world that is named recent invertebrates. That, of course, was done to discriminate it from the fossil invertebrates. But to your question, ants as a group have been around for, give or take a few million years, about 150 million years ago. Cretaceous, so fairly young group. And their great diversifications, like that of many other groups, flies and wasps and bees, for example, is related to the rise of vascular plants about 100 million years ago. We still don't know what the Proto Ant looked like. Some advances have been recently made. What do they look like? Do they look more like wasps? Or do they actually look more differently, more like ants? There is multiple lines of evidence, but we know they were around, but did not start diversifying and virtually exploding until all the trees, all the shrubs, all the flowers actually came around. And like a great coevolution started between these groups, and it's still existing.
We had a question about one of the ants on a few slides earlier, that you had an array of ants and one in the middle had a giant head. I didn't know if you could tell us what that was.
Tell me when to stop. The one in the middle? So what you see here is a group or a genus called Fidoli. You can see the name right there. So this is a group of ants that are somewhat unusual because they have a dimorphic cast, meaning they have 2 sets of workers that look very different from one another. Most of the workers, the so-called minors, will look something similar to this, And then the majors have these tremendously large heads. And actually, what they are used for, these guys mostly, is imagine fancy nutcrackers. These guys feed primarily on seeds. So the seeds are harvested by the miners that forage outside, bring the seeds, and these large-headed guys with their powerful mandibles will just crack the seeds so then they can feed them to the larvae. And in addition to that, they also defend the nest. Because they're so powerful, if intruders come, they're the first line of defense. But mostly, again, think of them as a nutcracker. That's what they use their big heads for. And their heads are so big because they house all the powerful muscles to move those very large, very powerful mandibles. Okay, so pictures are helpful, certainly. Oh, so how do we know something is new? How do we compare it? Do we use images of pre-existing species and then take images and compare them so we know it's new? Or do we do something else? Identifying insects, maybe with the exception of butterflies, is notoriously difficult by using images. We have to focus on characters. So when we find something new, again, every single one of us is expert in different fields. When I see an ant, I know what group it belongs to, what genus, right? I can tell you by just looking at them, and you can see how different they are. Maybe you don't know exactly what terminology to use, but this ant is different than this one, certainly different than this one. So we know what group. And within that group, we know to some extent what it's most closely related to, meaning that you have to have prior knowledge. And then, yes, we go back through the old literature. We don't look much at pictures. We read characters. This ant has 35 hairs on the back of its head, and they are all upright. The other species have those hairs all short, and they're all pressed to the head. Their appendages are that long. There is X number of segments in the antennae. So it is comparison, but it's not necessarily visual as it is actually quantitative. But images do help in some cases, especially with butterflies, again. And that is why many bird watchers have taken on butterflies, because you can use your binoculars and still identify them.
All right, those were some really excellent questions, and I think we're right on time. So I think Dr. Ivanov will be happy to stick around for some one-on-one questions and so forth. So I want to thank you all for coming out. South River Science Team, Center for Cold Waters Restoration, Wayne Theater, thank you all so much. And let's give Dr. Ivanov a round of applause for thanks one more time.
Thank you, guys.